In this lecture, we're going to talk about more special senses and hearing specifically. The ear actually houses two different of your special senses. It houses both hearing and equilibrium or balance. The receptors are mechanoreceptors, and different organs house receptors for each of those two different senses. The ear itself is divided into three areas. There's the external or outer ear, that's the part you can see, and then there's the middle ear or the tympanic cavity, which is where your eardrum resides, and then the inner ear is also referred to as the bony labyrinth. So in this diagram you can see the external ear includes the part of the ear that you can see plus part of your ear canal. The middle ear contains the tympanic membrane, that's your eardrum, and then the inner ear contains all of these different structures that allow you to hear and balance. The external ear is only involved in hearing. The structures of the external ear include the auricle and then the external acoustic meatus, or the auditory canal. The auditory canal is a narrow chamber in the temporal bone, and it's lined with skin, and then the skin also has ceruminous wax glands, and it ends at the tympanic membrane, which is also called your eardrum. The inner ear, the middle ear is called the tympanic cavity, and it's an air-filled cavity within the temporal bone. It's also only involved in the sense of hearing. It does not involve balance. In the middle ear or tympanic cavity, there are two tubes that are associated with the inner ear. The opening from the auditory canal is covered by the tympanic membrane or eardrum, and then the auditory tube connects the middle ear with the throat. This allows you to equalize pressure when you yawn or swallow. So if you've ever been on an airplane, for example, and the pressure in the cabin changes, you might feel like you need to yawn or something to pop your ears. And so the tubes allow you to do that. This tube is otherwise collapsed unless it opens up when you're equalizing pressure. In the middle ear, there are three little bones. There's the malleus, or the hammer, the incus, or anvil, and the stapes, or stirrup. And the function of these little bones is to vibrate. They vibrate from the eardrum, and it moves through the anvil to the stirrup to the inner ear. So those little bones, when they vibrate, carry the sound waves that caused your tympanic membrane to move into your inner ear. So here again you can see you have your external acoustic meatus. This is where sound enters the ear. It causes the tympanic membrane to vibrate, and then those vibrations are carried through the hammer, anvil, and stirrup to the inner ear. The inner ear is also known as the bony labyrinth, and it includes sense organs for both hearing and balance. It's filled with a fluid called the perilymph, and it contains a maze of bony chambers within the temporal bone, and those are called the cochlea, the vestibule, and the semicircular canals. So those three chambers in the inner ear, again, are the vestibule, the cochlea, and the semicircular canals. And those are all going to be involved with hearing and balance. First, we'll talk about the organs of your equilibrium ability. So equilibrium receptors of the inner ear are called the vestibular apparatus. The vestibular apparatus has two functions, or two functional parts, and it's because you are able to maintain equilibrium both in a static state, or not moving, or a dynamic state when you are experiencing movement. So much of this sense is housed in the vestibule, so that's kind of this big region here in the inner ear. There are structures for static equilibrium called the maculae, and these are receptors that are found in the vestibule. They report on the position of your head. So this sends information back to your brain, reporting on kind of what position your head is in, and it goes to your brain via the vestibular nerve. The anatomy of those sensors, or those maculae, they're hair cells, and they're embedded in something called an otolithic membrane. Otoliths literally translates as tiny stones, float in a gel around the hair cells, and when you move, it causes the otoliths to bend the hair cells. So the degree to which those hair cells are bent gives you the ability to determine where your head position is in space. So as you can see, again, this occurs in the vestibule, and here we have the otoliths. Those are those little stones, and they are embedded in this gel. The gel surrounds these hair membranes or hair cells that stick up from the supporting cells in the nerve. When the otoliths move, it causes the gel to shift and it can bend the hair cells in various directions. So in this diagram, you can see kind of a more dynamic explanation. So otoliths move relative to gravity or with gravity. And so when gravity changes, like you've tipped your head forward, the otoliths are no longer being pulled straight down, they're being pulled forward because you've tipped your head forward. That changes the gel that they are influencing, and that causes these hair cells to bend, and that message can get sent to your brain. The other part of equilibrium is dynamic equilibrium, and these receptors respond to angular or rotary motions. So there's a structure called the crista ampullaris. It's in the ampulla of each semicircular canal. So we'll talk about where that is in a second. And then there's dynamic equilibrium receptors in the semicircular canals. There are tufts of hair cells covered with something called a cupola or a gelatinous cap. And if your head moves, the cupola drags against a fluid called the endolymph. So this shows the ampulla, plural is ampullae, and that is where kind of this equilibrium that is dynamic is housed. So if you were to experience a spin or rotational force, first we'll talk about the structures, right? So the fluid is the endolymph, and then there's this gelatinous cap called the ampulla, and then there's these hair cells that are influenced by the movement of the ampulla. So if you are spinning, for example, here our direction of movement is spinning to the right, it causes the endolymph to flow over the ampulla, and that causes these hair cells to bend, and that allows your brain to detect how you are moving through space rotationally. So with dynamic equilibrium, the movement of the cupola stimulates the hair cells, and then an impulse is sent via the vestibular nerve to the cerebellum to process that information. Now we can move on to hearing. One of the organs that is associated with this is called the organ of Corti. It's located within the cochlea, and it has receptors. Those receptors are called hair cells, and they are planted on a basilar membrane. There's gel-like tectorial membrane that is capable of bending the hair cells, and then there's the cochlear nerve attached to the hair cells, and that transmits nerve impulses to the auditory cortex, which is in your temporal lobe. So your hair cells are going to collect the information, and then they're going to send that information via the cochlear nerve to your brain. So within the temporal bone, you have the organ of Corti, so that's here, and that is where sound information is processed, and then it's sent via the afferent fibers of the cochlear nerve to the brain. And here you can see within the organ of Corti, you've got the hair cells. Those are the cells that vibrate to different sound wavelengths or frequencies, and then they are embedded in this basilar membrane with these supporting cells, and they send their information to the cochlear nerve. So the way that hearing works is that they vibrate from sound waves that move the tectorial membrane. Hair cells are bent by the membrane, and action potential then starts in the cochlear nerve, and the impulse travels to the temporal lobe, and continued stimulation can lead to adaptation. So a sound wave is essentially a pressure wave, right? And because that's how sound travels through the air. And first, it hits the external ear, the pinna, and then the auditory canal, and then it strikes the eardrum. The eardrum takes that vibration from the air and transfers it to vibration of the bones in the middle ear. And then that causes the fluids in the cochlear canals to vibrate at different frequencies, and that stimulates the hair cells in the organ of Corti, and that allows that message to travel to the brain eventually. Hair cells are able to pick up different frequencies. So high-pitched sounds disturb the short, stiff fibers of the basilar membrane. And so those are receptor cells close to the oval window. And then low-pitched sounds disturb the long, floppy fibers of the basilar membrane. And so you've got hair cells for each of the different sound frequencies that you can hear. Hearing damage can actually occur if those hair cells that hear specific frequencies or vibrate to specific frequencies are damaged because they cannot be regenerated. So if you damage enough of the hair cells that are within the organ of Corti, you will lose your ability to hear anything that vibrates at those frequencies. So in this diagram, you can see kind of the different wavelengths of sound that you're able to detect with those hair cells. And so there's different frequencies. So high notes around 20,000 hertz all the way down to low notes around 20 hertz are all sounds that are within kind of the normal hearing range for people. But that ability can be lost again with hearing damage because each of those individual hair cells detects a slightly different frequency. And once a cell dies, it cannot be replaced.